And this is all about the central dogma. What's the first step in the central dogma? It's our understanding of how biology, and especially how genetics works. So what's the first molecule involved in the central dogma of biology? DNA. Okay. That's always a good default answer for a genetics class. DNA. Okay, what does DNA encode? RNA. And that's what we're working on right now, is the process of transcription. Proteins that read DNA and then produce an RNA molecule. And then what's the next step? RNA encodes proteins. And that's translation, and that's the next chapter. And for a long time, the idea of the central dogma was one gene, one transcript, that is a molecule of RNA, one protein. That is, one gene makes one messenger RNA molecule, makes one protein. Which is unfortunate for those organisms that have really small genomes and not very many genes, because if there's one gene to one protein, then how many proteins is an organism limited to making? Equal to the number of genes that they have in their genome. So if you have a little genome, if you have very small numbers of nucleotides in your chromosomes, then necessarily you can't make that many proteins. But it takes a lot of proteins to do all the things that cells do, and that's why today we're talking about alternative splicing. So this is part of transcription, but what if I told you that one RNA molecule could make 6 or 8 or 10 different proteins? That's what splicing does for us. DNA encodes RNA. What enzyme does that process? What makes a molecule of RNA? There's an enzyme, a protein, that does this job. It reads the DNA molecule, spits out RNA as it goes. RNA polymerase. Right? DNA polymerase makes DNA. That's when chromosomes are copied. RNA polymerase reads DNA and makes RNA. So after RNA polymerase makes the primary transcript, that should be a familiar word from the video I asked you to watch, transcription produces a primary transcript. In eukaryotes, us, then alternative splicing is. And that's what we're talking about today. So as RNA polymerase, the first step in RNA polymerase activity, transcription, is it has to figure out where on the chromosome to land, where it binds, to start reading the DNA and producing the messenger RNA transcript. The next step, this should be review. If it's not, watch the movie, is elongation. That is the connecting together of ribonucleic acids to produce the messenger RNA molecule. And then RNA polymerase has to figure out where to stop reading the DNA. So between where the RNA polymerase starts reading and stops reading, is called the coding region of a gene. So if that's DNA, RNA polymerase will make a messenger or an RNA molecule, the primary transcript, that's as long as the distance between where it starts transcribing, it being RNA polymerase. RNAP starts there, ends there, and produces an RNA molecule that's as long as the DNA molecule it just transcribed. What else do we call a coding region? What is it? It's a transcript of a gene. So this is the concept of a gene. Part of the chromosome, part of DNA, that gets transcribed by RNA polymerase and turned into a messenger RNA. So what's the easiest way to figure out where our genes are? How do geneticists functionally define what is a gene. We have huge chromosomes. We've got about 30,000 genes. There's a lot of nucleotides in between each of our genes. So how do we know where a gene starts and a gene stops? You pull all of the RNA molecules out of somebody's cell, and you line them up to the DNA sequence, and that tells you where the genes are. Because a gene will encode or produce a transcript after RNA polymerase reads the gene and makes RNA. That's the easiest way to figure out where the genes are. Collect all of the RNAs from a cell, and you see where their sequence corresponds to DNA. So the main questions that geneticists had to figure out is, how does RNA polymerase know where to start reading and where to stop reading? This is critical. That's what defines our genes. That's what defines the proteins that our cells produce. It's all about where RNA polymerase binds to the DNA and starts transcribing, and then where it lets go. You've already, hopefully, watched a part of a video that talks about initiation of transcription and elongation. Today we talk about splicing and termination. I'm not going to go through this because it was in the movie. The primary point I wanted you to take away from this part was nucleotide numbering. The +1 nucleotide is the first DNA nucleotide that gets read by RNA polymerase. Okay, so know the nucleotide numbering system. Elongation, the thing that I'm most interested in you figuring out and knowing what to do is to be able to look at the sequence of a coding strand of DNA, the template strand of DNA, and the transcript. And if I tell you one of the three, figure out which the other two are. So in this picture, down here at the bottom, which one of these is the RNA sequence? How would you know what's RNA and what's DNA? What? Yeah, you. Yeah, you. Over there. What? Right. Uracil is in RNA. Tablet?
I'm so sorry.
That's okay.
I left my iPad outside. Thank
We got
you
it.
so much. I can go in.
Yep.
It's fine.
No worries.
Thank you so much.
Yep.
I really appreciate that.
Have a better rest of your day.
Thank you.
Yeah, absolutely. So yes, U is how we tell what's RNA. So which of these two strands of DNA then is the coding strand, the dark blue or the light blue? What if I ask you which one is the template strand? So this is an important distinction. Right? If DNA is double-stranded, RNA polymerase only reads one of those two strands as it moves along the chromosome and reads the DNA sequence and spits out RNA sequence. So the one that's base paired to the RNA is that the coding strand or the template strand? This is the template. So you can think of the template is that's the molecule of DNA, the strand, that RNA polymerase is actually reading, and it's making base pairs with the DNA. It's base pairing RNA nucleotides with the DNA template. That's how it makes the copy. That makes the other strand the coding strand. What's useful about the coding strand? It's not the one that actually gets copied, transcribed. It's the other strand. What's its sequence going to be if from left to right, that top molecule, the coding molecule, the navy? Left to right, it's going to be the complement of this sequence, right? So it's going to be T base pairs with A base pairs with T, and so forth. What does that sequence look like? So the coding strand, what I just wrote in there, ATC, GGG, etc. What is that? The the same sequence as the RNA, except it's got T's instead of U's. So that's why it's called the coding strand. That strand of DNA has the same sequence, except for replacing T's with U's, as the transcript, the messenger RNA molecule. So the coding strand is always going to have the same sequence as the RNA molecule. The template strand is the one that the RNA polymerase is actually copying from. Any questions about this? So that's what I was hoping you took away from the video for today. Quick summary. Okay. So in eukaryotes, multicellular organisms with nuclei, there are three things that happen to RNA molecules after transcription. So after RNA polymerase has started to transcribe, three things happen. The addition of a 5' cap, so at the very 5' end of the messenger RNA molecule, again, by way of review, 7-methylguanosine gets added to the 5' end. That's one step in processing, the 5' cap. What happens at the 3' end? Don't just say poly A tail, because I can read that. What does that mean? What happens down here at the 3' end of an RNA molecule? What does it mean to have a poly A tail? What does poly mean? Multiple, yeah. Don't tell me anything about no parrot. Oh, I know, I'm sorry, it's Monday. So it's a multiple A tail. So the end of every processed messenger RNA molecule in eukaryotes has had a bunch of A's added onto its 3' end. So that's another step. That's two of the three steps. The addition of a molecule on the 5' end, 7-methylguanine cap, addition of additional nucleotides to the 3' end, poly A tail, and then there's splicing. And that's the removal of introns. Do you have a pretty good sense of what an intron is? You've probably heard about them before, maybe. You've at least heard somebody say an intron, right? What's an intron? What's its job? What's its role? What? Say that again. Say it louder. Yeah, so there are lots of jobs that introns do. Sorry, I misheard you. I thought you said it takes up space, which it does. I mean, introns do have jobs. Introns, the DNA sequence that are in between exons, do things for us. But in general, they're just parts of the mRNA transcript that get taken out, which is kind of strange. So the goals for today: describe what splicing is, predict splicing patterns, which hopefully will make more sense in a minute if you don't know what that means already, and then we'll talk about termination of transcription as well. So, what is splicing? What are introns and exons? How do you know what an intron and an exon are? So it's typical for geneticists to diagram exons as boxes and introns as just straight lines. So here we're looking at the sequence of an RNA molecule from 5' to 3'. And I'll just annotate exons with E's. So we've got 3 exons here, E1, E2, and E3. And we've got 2 introns, intron 1 and intron 2. How do I know that there are 2 introns if I'm a geneticist and I'm studying an RNA molecule? It's what's in between the exons. How do I know what are the exons? I'm just trying to figure out the definitions here. So somehow scientists, geneticists in particular, can take a single RNA molecule, just the sequence of nucleotides, 5' to 3', somehow we have to figure out where are the exons and where are the introns. How do we do that? Okay, so I heard start and stop codon, the words. So somebody said start and stop codons. That has to do with translation, which is the next chapter. Those codons are going to be in exons. So I'm just going to diagram them here just because they came up in conversation. I'm going to show you that start and stop codons are part of exons. They'll be found inside exons. So that could tell us that there are exons there, containing the start and the stop codons. What else? Yeah?
Would you just compare the fully processed RNA to the original DNA and look at which parts are not in it?
Okay, so we know that introns are parts of messenger RNA molecules that get taken out. That's the process of splicing. So what if I show you that after splicing, we have this molecule. It's got a poly A tail. It's got a 7-methylguanine cap at its 5' end. And it's got exons 1, exon 2, and exon 3. Those DNA sequences together. And there's nothing in between the 3 exons. So if you line up the DNA sequence or the initial pre-mRNA sequence shown on top, and the fully processed or messenger mRNA on the bottom, how do you define what an intron is? Exactly what you said. It's everything that's been taken out. All of the messenger RNA that was in the pre-mRNA, intron 1 and intron 2, if you don't find that when you look at the sequence of the fully processed, capped splice, poly A-tailed messenger RNA, that's how we define introns. It's the DNA that gets taken out of a primary transcript. So we actually functionally define introns. We don't know the sequence of the introns when we start. We just take messenger RNA and the pre-mRNA and we compare their sequences. What's in the pre-RNA that's not in the messenger RNA is an intron. There are particular sequences, though, that are part of introns. Does anybody remember? There are nucleotides at the start of every intron and the end of every intron that are almost always the same nucleotides. There's 2 of them. So the first 2 nucleotides of almost every intron and the last 2 nucleotides are always the same. Doesn't matter what species you're talking about, doesn't matter which gene. So the first 2 are always G and U. What are the last 2? Does anybody remember? A and G. Ah, you knew that, yeah. That's fine. There's also a third nucleotide that's important, or a third set of nucleotides. So there's the 5' splice site. That's GU. There's a 3' splice site. That's AG. And then in the middle of those 2, there has to be something called the branch point, which is just a single A. And there could be any number of nucleotides in between, literally any number, tens, hundreds, thousands of nucleotides in between the 5' splice site and the branch point, and between the branch point and the 3' splice site. So introns can be huge. Introns can be bigger than the gene itself, which seems sort of stupid from the cell's perspective, because what's the point of introns? Because RNA polymerase is going to go through the trouble of transcribing the entire gene, all of its exons and introns. Then what's it going to do to the introns? Not RNA polymerase, but after splicing, what happens to the introns? They get taken out. What's the point of introns? It's like extra nucleotides in your chromosome. They get processed by RNA polymerase, and then they get taken out and thrown away. Weird. But nevertheless, it happens. And again, yeah, some introns are bigger than the gene itself. So it's very strange. Some wonder why our cells do this, and hopefully by the end of today you'll have at least a little understanding of why splicing might be important. So with that knowledge, I guess we could also write in the same sequences here. So what's going to go at the 5' end of intron 2? The first two nucleotides? This is something you should memorize. GU. So I'm just asking you to say it because you know that helps improve memory if you say it. And then the branch point is A, and then the last two nucleotides are AG. So that's a DNA or an RNA signature set of nucleotides that you can look for to try to find introns. So initially, this was the one gene, one messenger RNA, one protein model. We've got one gene, it's got 3 exons, 2 introns. We've determined that it's got 2 introns because when we look at this final messenger RNA molecule, those sequences aren't present in that RNA sequence. Then we saw things like this. We being geneticists, definitely not me, this is well before my time. We also saw things that looked like this. So that's another messenger RNA molecule. So this first one's mRNA1, call this one mRNA2. And then we also see types of RNA that look like this. Bless you. Okay. So the second RNA molecule has exon 1, exon 2, exon 3, just like the messenger RNA 1. What's different about 1 and 2? It's got an intron. So this messenger RNA molecule still has intron 1. What happened to messenger RNA 3? What doesn't it have? It doesn't have exon 2. I mean, like normal, it doesn't have intron 1 or intron 2. But what happened to exon 2? It's not there. So these represent 3 different patterns of what we call alternative splicing. And the point of alternative splicing is that when scientists started going into cells and taking out the RNA molecules, they noticed that when a gene gets spliced, when its introns get removed, the cell does not always splice every piece of messenger RNA, that is, every single RNA molecule in the same way. So sometimes, as in the case of messenger RNA molecule 1, it's fully spliced. All of the introns get taken out, all of the exons stay. So you get exon 1 next to exon 2 next to exon 3. Messenger RNA 2 represents intron retention. So not every intron always gets spliced out. Intron retention. Okay. Messenger RNA 3, when you skip an exon, is not surprisingly called exon skipping. Colloquially, in conversation by geneticists. So we've got fully spliced, you take out all the introns, no introns get retained. We've got intron retention. Sometimes you'll see that messenger RNAs don't have all of the introns removed, and sometimes exons get removed, exon skipping. And you can have all sorts of versions of this going on. And a lot of genes have a large number of exons, not just 3. I can't fit more exons with my style of handwriting on this slide, so I choose a simple example. Lots of genes have 20, 30, 40 exons, which means something like 19, 29, 39 introns. And then I could ask you, I won't, but I could ask you something like, how many possible combinations of splicing do you have if you've got 40 exons when you can have intron retention and exon skipping? So you make a crazy number of RNA molecules after splicing from a single gene. So this is getting back to that point I made earlier. One gene can encode tens, hundreds, thousands of different transcripts and make tens, hundreds of thousands of different proteins from the same gene. Presumably that's the point of why eukaryotes, what we might call higher organisms, splice their genes. It creates diversity. One gene can make loads of different proteins instead of just one. Presumably alternative splicing is what creates diversity and leads us away from the one gene creates one protein model into one gene creates a huge number of proteins.
What molecular structure determines what gets pulled out? Like, I mean, because this is like so small, it's inside the cell, there's not like a brain, you know, or something that's thinking.
Right. So inside the cell, it's a fabulous question, and hopefully where we're going to head next. Yeah. How does the cell determine, how do cellular mechanisms determine when introns get retained, or when they get spliced out, or when exons get retained? So hopefully, intellectually speaking, fully splicing is kind of intuitive. So we have proteins in our cells that recognize the splice sites. And they physically bring those splice sites into opposition, that is, next to each other. And that catalyzes the actual reaction of removal of the intron. And I'll leave that to a biochemistry class if you want more details. How the intron actually chemically gets removed from the RNA molecule. So can you imagine a situation of what might be happening given that proteins are involved in removing introns? It makes sense that things be fully spliced. Every intron gets taken out by these proteins in every messenger RNA molecule. Why might introns get kept in sometimes? Or how, not why? What mechanism might cause some introns to stay in the RNA molecule? Your proteins are involved in this process. So what sort of a situation inside a cell might cause some RNA molecules not to get spliced? What are those proteins that are splicing doing? They're somewhere else splicing some other RNA molecule, and you just happen to get missed. So it kind of depends in part on the concentration of those proteins that do the splicing. Are there enough inside the cell to handle all of the transcripts that are inside the cell at that point in time? So if the cell has too few of these splicing proteins relative to the number of transcripts, sometimes introns might not get spliced out. So that might explain intron retention. Sometimes not every intron gets spliced out. What about exon skipping? This might be conceptually easier. So fully splicing, I think, makes sense. Every intron gets taken out. Cell doesn't have enough proteins to do the splicing at any one point in time. Maybe introns get retained. What happens during exon skipping? How does a cell remove a couple of introns plus an exon at the same time? These proteins that do the splicing recognize the splice sites, GU, AG. So how could a cell, or the proteins inside the cell, take out two introns and an exon at once? Here's a GU. Where's an AG that will cause exon 2 to get spliced out? Right there. So if a cell accidentally or purposefully, like you said, the cell doesn't have a brain, so it's hard to tell which, splices the GU from intron 1 to the AG of intron 2, then you remove both the introns and the exon that's in between at the same time. I'm going to let that sink in for a second. If you've got a question, do please ask or raise your hand and then ask before we move on. The simple point is cells do all of these processes to most of their genes all the time. So I just want to give you one example of why alternative splicing might be important. So here are a bunch of different fish. They're from very closely related species. These are all cichlid fish, and these are all found in lakes in Central Africa. So cichlid fish, these are from which lake? Lake Malawi. You find the same sorts of fish in Lake Victoria. They're all close relatives. They're sister species. If you look at their genes, they're almost identical. So the evolutionary biologist in you, I know it's in there somewhere, should ask, what? Presumably a lot of you are going to be heading into Biology 105 at some point, not all of you. What causes all this diversity? If I showed you and I didn't tell you first that these were all really closely related species, would you guess that they're closely related species? What's different about them? Yeah, pretty much. Everything. Size, shape, so mouse size, body structure, color. They have vertical bars, they have horizontal stripes. Mostly it's color and pigment patterns, body size. Huge amount of diversity. But these species are almost genetically identical. Not really genetically identical, but close. So how is it that despite being very similar in DNA sequence in all of their chromosomes, how do these fish make so many different color patterns? These authors of this paper that I'm presenting to you briefly, just a few figures, knew this about zebrafish. And this is a wild type zebrafish. Guess why it's, never mind. It's called a zebrafish because it's got stripes. They found a mutant. It was already known that there was a mutant. So if you get a mutation in a particular gene called hagoromo, which I have been told means dress of the gods in Japanese, if you have a mutant, what happens to the pigment? What happens to the zebra stripes? They get disrupted. So you don't get these nice, clean black stripes that go from the anterior to the posterior of the fish. That pigmentation pattern gets messed up a little bit on the side of the fish. So mutation to this gene causes this phenotype. And so the authors of this paper, they were studying the cichlid fish, all these different colors and patterns. You've got vertical stripes and horizontal stripes, thought, maybe in all of these fish from Africa, there are mutations or different alleles anyway of the hagoromo gene, and that's what causes different striping patterns. So when they went in and they took out the sequence of the hagoromo gene from the cichlid fish, this is what they found. So this up here is the structure of the DNA. When by structure, I mean this is a molecule of DNA from left to right. This is a representation of the chromosome. And there are 9 different exons of the hagoromo gene. So each exon is just represented by a single vertical bar. And you can see that there are some, here's the scale bar. So how big is the entire gene? How much DNA gets transcribed by polymerase, RNA polymerase? And I'm going to try to redraw that scale bar so I can kind of, that maybe 50 kb, something like that. So it's 50,000 base pairs long, this entire gene. These exons are drawn to scale. So out of 50,000 kilobase pairs, the exons are tiny in comparison. So down here they've blown up this really compacted region that has exons 1 through 5, so we can see them in their relative sizes in a little bit more detail. And they just wanted to know, they had this hypothesis. Remember, we're talking about splicing, we're talking about pigment patterns. The authors thought maybe the way that these different fish have different pigment patterns, different striping patterns, is maybe this gene gets spliced differently in every one of the different species. And that a different messenger RNA molecule then gets produced for every different species from this same gene. And maybe that's what causes the different striping patterns that are found in all these species of fish. So what did they have to do? They had to actually take, well, it's rough life. They had to go to Africa and scuba dive and collect all the fish. Sometimes it's nice to be a biologist. Then they just extracted the RNA from the tissue samples they got, and they just looked at the sequences of all the RNA that came from this gene to find out where are introns and where are exons. And they found that these were the 9 most represented splice forms of this gene. So a hagoromo gene in different fish were most often found in one of these 9 varieties. So at the top, we've got the fully spliced version, what you and I might normally expect a gene would do. It gets all of its introns removed and all 9 exons get retained. No introns left, no exons spliced out. What else did they find? We've got intron retention, so every time you see a gray bar, there was an intron that got left in. And we've got some exon skipping. So who can find a version that's got an exon that's been removed? We've got 9 exons. So which number, 1 through 9, is missing an exon? Okay, so 9, we've got exons 1, 2, 4, and 5, and that's it. So what happened to exon 3? Got spliced out. What happened to exons 6, 7, 8, 9? They got spliced out. So they did find a pretty big variety of different splice forms. So alternative splicing of this gene in this species is happening. That's the bottom line. It takes a long time for me to say simple things sometimes. Right. I don't like to deal always in generalities. Sometimes I like to show you that the data actually suggest what I'm trying to convince you is true. Alternative splicing happens. These authors went on to do some other analyses to try to prove that these different splice forms were involved in different pigmentation patterns. The conclusion was a little bit vague. So I can't tell you whether or not the different versions of this messenger RNA, the different versions of the hagoromo gene, do cause differences in banding patterns. But it was an interesting hypothesis. So any questions about alternative splicing? RNA polymerase makes 1 molecule of RNA. It's got all of the introns and all of the exons when it transcribes the gene. And after that, splicing proteins do their job. They either take out all the introns or they take out some of the introns. Sometimes they take out exons as well. It produces diversity, and presumably that diversity is useful. At least some of the time. So that's the concept of alternative splicing. Okay. So how does an RNA polymerase know where to stop transcription? Talked about initiation, that was in the movie for today. Got 3 different steps of RNA processing: capping, tailing, and splicing. Now we've got to figure out how a polymerase knows where to stop reading the DNA and producing RNA. It differs between prokaryotes and eukaryotes, which is pretty common. In eukaryotes, there's a particular DNA sequence that encodes a structure that makes RNA want to base pair with itself. So it's important to remember that RNA is a single-stranded molecule when it is produced by RNA polymerase. RNA polymerase just makes a single strand. But RNA is a nucleic acid, and so it likes to base pair with things when it can. And it turns out that this particular sequence that's the end of every prokaryotic gene causes the RNA molecule to fold on itself, to base pair with itself, to make a little hairpin, like that. There it is in actual detail if you want to know something about the RNA sequence. I don't want you to know anything about the RNA sequence. But this is what happens. So the G base pairs with the C, A with U, C with G, and so forth. And this physical structure, a hairpin that's in an RNA molecule, is sort of like the key to unlock RNA polymerase. So this hairpin structure actually physically interacts with RNA polymerase, the protein, and makes it fall off of the DNA. It's like there's a little slot. You could think of it this way. It's like there's a little slot inside RNA polymerase And when this key, the hairpin, gets inserted into the slot, that makes RNA polymerase fall off of the DNA molecule, stops transcribing. That's how termination occurs in prokaryotes. Bit different in eukaryotes. So we don't use hairpins, but we still use an important DNA sequence that tells RNA polymerase where the end of a gene is. In eukaryotes, it is, not surprisingly, a particular DNA sequence, AAU-AAA, which is also called the polyadenylation signal sequence. So this, I would like you to memorize, AAU-AAA, polyadenylation sequence. That signals the end of a eukaryotic gene. And that sequence gets transcribed. So on the right side here, I'm drawing an arrow that points at that sequence as it's transcribed by RNA polymerase. So RNA polymerase actually reads across the DNA through the AAT in DNA, AAA. So RNA polymerase makes AAU-AAA at the end of the transcript. That's that little red bit. And when that little red bit, AAU-AAA, interacts with a particular protein, that is what tells that protein to cut physically the RNA molecule. So about, as it says here, about 20 nucleotides pass the AAU-AAA in the RNA molecule. So we've got some sort of nucleic acid sequence, then there's AAU-AAA, then there will be about 10 to 20 nucleotides. This protein complex cuts the RNA molecule, and that's its new 3' end. And this is the point at which poly-A polymerase, an enzyme, does the tailing. It comes in and it just adds a bunch of A's to the end of the RNA molecule. And now we have a fully processed, capped, spliced, and poly-A tailed messenger RNA. So there are particular DNA sequences, summary, particular DNA sequences in prokaryotes and eukaryotes that cause termination. In prokaryotes, that sequence forms a stem loop, which causes RNA polymerase to stop transcribing. In eukaryotes, AAU-AAA gets recognized by a separate protein. And as soon as that protein sees that sequence being produced in the RNA molecule, that causes the RNA molecule to be cut about 10 to 20 base pairs beyond the AAU-AAA. And that's the 3' end of a eukaryotic transcript. So everything I'm telling you today, functionally, when we get down to doing quizzes, exercises, exams, is how can you recognize, or can you recognize, and by what data do you recognize an exon, an intron, where a gene would terminate transcription, and how does it get modified? So capping, splicing, polyadenylation. Ultimately, I'd love to be able to just give you a DNA sequence and say, here's a DNA sequence. Show me which part of it gets transcribed. Tell me which parts of it are introns and exons. Write out what an actual fully processed, capped, spliced, and tailed messenger RNA molecule would look like. And I've got, yeah, I know. This is real genetics now. So this is a summary. I've created a Google Classroom PDF exercise that does exactly what I just asked. I'm never going to ask you to do all of that on an exam, although I might ask you to do it in separate questions and not necessarily in that order. And it looks something like this. So this is the exercise. I'm about to send it to you via Google Classroom so you can have a PDF of this. It's just the sequence, it's the DNA sequence coding in template strands. They're the second and third lines. So it's a DNA sequence. right there. And then on this exercise, there are a number of questions. Can you identify where a transcription would start, where a transcription would stop, where are the introns, et cetera. So we're going to work through this together. I'm not asking you to do it on your own necessarily. You can work in it in groups if you want. Try this by yourself in groups. Come back next class. Hopefully I'll be here too. I don't think they allow me to Skype in from a jury if I'm on a jury. So that's, yeah. I don't know if that's good or bad. And so we'll work on that both next class and then in subsequent classes. So this one exercise, you won't turn in yet. I just want you to work on it and come asking questions about how to interpret what I'm asking you to do, issues you ran into, and so forth. We're going to keep working on this same exercise through the next chapter as well. So we're going to do transcription. We're going to figure out from that DNA sequence what messenger RNA gets produced. And after that, we're going to take that messenger RNA next chapter when we do translation and figure out how we know what protein gets encoded by this messenger RNA. To close, not quite there yet, close. This is the question that we're dealing with next chapter. We're not to translation yet, but I just wanted to put this thought in your mind. Here is a messenger RNA sequence. Tell me what protein is produced by this messenger RNA. That's the goal of the next chapter, and that's why we're working on this PDF exercise. Lastly, I'd like you to take some time. I'm going to launch, I haven't launched it yet, I'm about to, this Socrative quiz, just for attendance, and it's asking you your opinions about how last week went. So because we're coming up to some more times during the semester when I might be absent, I just want your feedback on, you liked it, you didn't like it, so I've just got 3 questions, I think, on the survey. It's more of an attitudes, opinions. Tell me what you thought. No scientific content. So please do this today, and then I'll hopefully see you on Wednesday.